Good morning, my dear students. Today we are discussing about one more topic in genetics. That one is gene therapy. What is the gene therapy and what is the importance of the gene therapy? And now we discuss. So here, gene therapy it related to the this is the one of the part of the Human Genome Project. We already discussed about the Human Genome Project. So what is the importance of the Human Genome Project and what are the different types of steps are involved in the Human Genome Project and what is the uses and importance in our daily life? We are already discussed about that. So same like the gene therapy means simply gene therapy is a experimental techniques. It is a experimental techniques. So that the experimental techniques that uses in gene to treat or prevent a disease means so treatment and nowadays different types of treatments are occur. The treatment is with the using of the drugs, with the using of the medicine, with the using of the homeopathic medicine, with the using of this different types of sources are there. But one of the sources is the with the using of the genes, with the using of the genes. So we are treat them one disease, especially with the using of the disease, with the using of a genes, a treatment of a one disease is known as the gene therapy. So gene therapy means simply it is a treatment of a disease with the help of the genes. That is a gene therapy. So here it is the use of the DNA. It is a pharmaceutical agent to treat a disease. So in the genes, which one part? DNA. So in the genes, what is the gene therapy? Simply with the help of the genes, treatment of a disease. In the gene, different types of parts are involved in there. Especially DNA involved in the pharmaceutical agent to treat a disease. It derives the name from the idea that DNA can be used the supplement or alter gene with the individual cells and therapy to treat a disease. So individually, the DNA is involved in the treatment of a disease that is called as a gene therapy. Here, several approaches in their gene therapy. What are there? Number one is replacing of the muted genes that causes the diseases with the healthy copy of the genes. So mutation means simply sudden changes occurs in the gene line. Sudden changes occurs in the genetic line. So here sudden changes occurs means it is the healthy gene converts to diseases. So here that one is replaced with the gene therapy. And the next one, inactivating or knockout muted gene that is functioning improperly. So which one gene is involved in the functioning improperly working, or knockout genes or inactivated genes? Which one genes? That one is eliminated from our body. So that eliminated from our body, replaced with the gene therapy. And the next one, a third one is introducing a new gene into the body, to help the fight a disease. So our body suffered with one type of disease. That disease is a cure with the help of the new gene. That new gene is introducing in our body. That gene is fight with the disease and decreases diseases. And the next one, this is about the gene therapy introduction. So in here in gene therapy, although gene therapy is promising to treat the option of the number of diseases, so in here not only one disease, number of diseases are there, including inherited diseases, some types of cancers. And the next one, inherited is some types of cancer, certain viral infections treat with the gene therapy. So here the technique remains risky and still under study to make sure that will be safe and effective. These are the safe and effective. So the gene therapy currently only being tested the treatment of diseases that have no other cures. So here the gene therapy first concept study in the 1972. So the gene therapy studied in the 1972 with the authors using the occasion of before the romancing the gene therapy studied in humans. So in 1972, study about the gene therapy. Simply, the first FDA approved gene therapy experimentally United States in 1990, when Ashanti De Silva was treated the ADA SCID Since then, over 1700 clinical trials occurs with the especially ADA and EFD, ADA and SDI. And the next one, the 1700 clinical trials have been conducted using the number of techniques of the gene therapy. 1700 clinical trials occurs. Clinical trials means before the drug is entering in market. So that one is a clinical trials occur. The clinical trials involved in the four steps, 1, 2, 3, 4 steps are there. So the 2, 3 is known as studied in animals, 3, and the next one, fourth one step is studied in human bodies. So that one clinical trials. Although early clinical features as a many dismisses gene therapy over the hyped and clinical successes in the especially 2006 to 2011 is new optimized promises of gene therapy. Number of clinical trials studied on the gene therapy, some are the rejected and some are successful, and finally we get the 2006 to 2011 we get the successful results in the gene therapy. So these including successful treatments is the patients with the renal diseases, is the labor congestional enumeration, X-linked diseases, and ADA, SCID, and then leukoproteins, chronic leukemia, and acute leukemia, and multiple myeloma, Parkinson's disease. These all type of disorders are studied with the help of the gene therapy. So these are all are cured with the gene therapy only. So the next one, approaches in the gene therapy. What are the different types of approaches? So first one is the gene therapy is designed to introduce the genetic material into the cell and compounds to the abnormal genes to make a beneficial protein. So how to treat the one disease? Simply, gene therapy is designed introducing the genetic material. So which one cell is suffered with the disease? Which one cell is suffered with the disease? So the genetic material is transferred into that cell. The genetic material transferred into that cell. And the next one here, genetic material transferred into a cell. So the composite abnormal genes are made into the beneficial protein or beneficial genes. And muted genes means abnormal genes. So muted genes causes the necessary problems to faulty missing, or gene therapy may be able to introduce the normal copy of gene, restore the function of the protein. So which one cell is the muted? So which one cell is the muted or abnormal cell? Abnormal cell converts to simply normal cell. So and normal cell with the help of the gene therapy. And the gene, a gene that inserted in directly into the into a cell, and into a cell usually does not functioning, and instead a carrier called is a vector. So here one cell is not working. So the into that cell transfer the one more cell with the help of the vector. Vector is nothing like a carrier. Vector nothing like a carrier. The carriers are transfer the new genes into them, into the which one cell is affected, which one cell is the muted, into transfer to the vectors. So this genetically engineered and to deliver the gene. So the certain viruses are used to vectors because they are deliver the new genes by infected cells, and virus modified that cannot causes the diseases. So in that moment, the transfer of the genes from outside of the body to inside of the body, or inside of the body to inside of the body. So here at that moment new genes are introducing. At that moment there is no chance for the formation of the new disease with the help of the gene therapy. And the next one, some types of viruses such as the retrovirus, and integrated the genetic material means new gene is into the chromosome of the human cell. So retrovirus into introducing the human cell, other viruses such as the adenovirus introducing the DNA into the nucleus of the cell, and the DNA is not integrated into the chromosomes, but chromosomes are introducing into the new cell, but not DNA not integrated into a chromosomes. And the vectors are the injected into the given intravenously. How to introducing the new cells into our body? Through the IV route, intravenously directly into the specific site of the tissue also, and taken up by individual cells. So this is about the approaches in the gene therapy. And the next one here, the approaches how to introducing the new cells with the help of the two methods are there. One is the in vivo gene therapy and ex vivo gene therapy. Simply in vivo means inside of the body, ex vivo means outside of the body. So inside of the body means what is the in vivo gene therapy? Genetic material introducing directly into the body of the affected person. Which one person is affected with the disease? So simply directly the genes are introducing into the body, like this one method. This one method. So this is the outside of the body directly inserted into the inside of the body. So ex vivo gene therapy, cells are extracted from the body. First of all, cells are extracted from the body, human body, and placed in the laboratory. Then that one is a modified and then introducing into the inside of the body, introducing to the inside of the body. Which one person is affected with the disease? That is called as a ex vivo. So ex vivo means this is the ex vivo. First of all, cells are collected from the body. Cells are collected from the body. Cells are collected from the body, and the next one here, laboratory condition, new genes are introducing into here, and that one cell is again inserted into the body. This is outside process is known as a ex vivo gene therapy. These two gene therapies are involved in the treatment of the disease. And the next one, types of gene therapy. So types of gene therapy is a different types of gene therapies are there. That one is classified as a two types. So mainly two types. That one is the first one is somatic gene therapy. Second one is a germline gene therapy. Somatic gene therapy and germline gene therapy. So what is a somatic gene therapy? Somatic gene therapy means these in the somatic gene therapy, the therapeutic genes are transferred into the somatic cells or patients or the body. So directly the genes are transferred into the body is known as the somatic gene therapy. Any modifications and effects will be restricted on the individual patients only and will not be inherited by the patient's offspring or the later generations. So this is about the modifications or effects restricted in the individual patients only. So this one is not inserted into the not inherited by the offspring or the next generations. And somatic gene therapy represents the mainstream line of the current basic and clinical researches. Will therapeutic DNA transgenes used to treat the disease of the individual treatments occurs with the somatic gene therapy. And germline gene therapy means, so germline gene therapy means here if the germline gene therapy, germ cells or the sperms or eggs are modified by introducing the functional genes like IVF. So like IVF, and the next one infertility conditions. So nowadays test tube baby centers, these are all are the follow the germline gene therapy. So these are the integrated into their genomes, then would allow the therapy heritable and passed out to the in the latter generation. So with the help of the IVF, with the help of the test tube babies, next generations is developed. But in the somatic gene therapy, there is no chance for the next generation develop. And here although they should be there, they highly effective and counteracting the genetic disorders, hereditary diseases. Many uses occurs with the applications occurs with the germline gene therapy. These two are the gene therapies are involved in there. And the next one more therapy is the vectors in the gene therapy. Vectors also involved in the gene therapy. So what are they? So vectors means the gene therapy utilizes the delivery of the DNA into the cells. Simply delivery of the DNA into the cells, which can be accomplished by the number of methods. So they are majorly two types only. So one is the viruses and the second one is the non-viral methods. So viral methods and non-viral methods. So what are their viral methods? So all viruses bind to the host and introducing the genetic material into the host cell, part of their replication cycle. So first of all, the viruses act as a vectors. So the viruses filled with the gene therapy techniques and introducing the new genes. Then that one is transferred into the host cell. In the host cell develop the new genes. So that is called as a viruses gene therapy. So there are the has been recognized by the possible strategies and for the gene therapy, and by removing the viral DNA and using the viruses vehicle deliver in the DNA. So number of viruses has been used the human gene therapy. What are the retrovirus, adenovirus, lenterovirus, then HSV virus, vaccinia, pox virus, virus, adeno associated virus. These different types of viruses are used as a vectors. And non-viral methods can be presented with certain advantages over viral methods, such as a large scale production purpose and low host immunogenicity conditions. We are prefer the non-viral methods. Low host immunogenicity, low immunity power in the cells. At that moment, we are choose the non-viral methods. So this is about the gene therapy and importance of the gene therapy. And here some limitations also they occur. So what are their limitations? So different types of limitations are involved in there. So what are their limitations? Now we discuss.